Hello and welcome. I'm Kevin Oslins, the Director of Enrollment at Clark Summit University. Here at Clark Summit University, our mission is to develop Christ-centered, career-ready graduates. As part of that mission, the Bible is at the core of everything that we do. In fact, every single one of our undergraduate students receives a double major in biblical studies and their chosen career major. So how does that work when it comes to a field like biology? At CSU, we believe that God created science, and therefore the more we understand science and biology, the more we will understand the God of all creation. We recently invited Dr. Lightner to share about what the biblical book of Genesis has to do with science. Dr. Lightner has received her doctorate in veterinary medicine from The Ohio State University. She has 27 major publications with research interests in genetics, adaptation, and speciation with a focus on birds and mammals. She does literature reviews and data mining to answer questions about how God's created kinds diversify and speciate. She has written for Answers in Genesis, Creation Ministries International, and published in the Creation Research Society Quarterly. Dr. Lightner did collaborative research with world-renowned taxonomist John E. Alquist, who studied birds. And we are thrilled to be giving you access to Dr. Lightner's insight in this webinar today. But before we hear from her, you're going to get the chance to hear from a recent CSU biology program graduate who is now earning her master's in biomedical sciences to serve Christ in the field of medicine. We're excited to see how God uses her through her passion for science. After that, you'll hear from our featured guest speaker today, Dr. Lightner.
Hi guys, my name is Andrea Sanchez. I am a junior here at Clark Summit University. I am a biology major, and you guys are probably watching this video because you are interested in the biology program, which, let me tell you, it is a great program and I totally recommend it. It has been such an amazing experience just being able to be a part of this program. You get to do the labs, you get to do TA work once you're in your second year of this program. And you are also just learning all of the sciences, and if you love science like me, well, great. But as you learn all these sciences, you will also be getting a biblical perspective. You will be taught by Mrs. Wright how God is working through all of these sciences, how His hand is within each and every single aspect of creation. And so I totally recommend it, and if you guys want to learn more about this, just reach out to us and we will totally get back to you guys.
Welcome to Biology at Clark Summit University. My name is Professor Wright, and I teach biology and chemistry here at the school. If you are looking at this video right now, it's probably because you have an interest in the sciences and you are curious to see what you would be able to do with a biology degree from Clark Summit University. I designed the program so that it would cover all bases. I have students that are interested in going into medicine, which a lot of students think of, but also going into lab work, going into working with animals, more environmental. So it's more of a broad type of biology program. It's general biology is the way I emphasize it. So during the time that you're here, you get a little bit of everything. So it gives you a chance to test the waters to see what area of biology you're most comfortable with, which area you are more passionate in, so that you can better choose your next step, which would definitely be grad school or med school. All biology students, yes, we know we're going on from here. One of the primary things that I think helps my students here at the school is we have two things that we really emphasize, and that would be the need for observation hours. If you're looking at different grad schools, they're going to require so many hours of observation, and so I work with each individual student on what they really prefer, and we try to find placements for them in that area so that they can get their hours in. But I also have a teacher's assistant program. This gives the students an opportunity to really find out what it's like to work in a lab, to work behind the scenes, to set up labs, tear down labs, take care of all the hazardous waste, take care of prepping solutions, keeping cultures intact, and working with other students. They help run the labs as long as they have completed at least a minimum of 1 year with me. Part of their program is they are required to work at least one semester after that, and that gives them a chance to really apply what they're learning in their lectures, and it also gives them something that's going to set them apart when they're looking for those positions in grad school. It's very competitive going on, and so you want to be able to set yourself apart what makes you different. And if you can show them that you have experience actually running labs, actually working with other students, it's really going to help you in those placements. Here at Clark Summit University, you're not only going to get a strong science background, but you're going to dovetail that with your Bible classes. You will get a biblical worldview that will clarify a lot of what you're learning in those science classes. There's a huge need for Christians in the scientific community, and so that you can stand on your own 2 feet and know why you believe what you believe. This is something that is unique to a school like Clark Summit University. If you have any other questions, you can contact us through the website, or you can ask for my email directly, and I'd be happy to answer all of your questions.
Well, hello and welcome. I'm Jean Lightner, and I'd like to talk to you about how the history in Genesis promotes an accurate understanding of biology. I'll be highlighting the Creation Research Society's eKind's predictive success. Now, to start off, maybe we should find out what eKind's is. eKind's is the name of a research initiative by the Creation Research Society, and it stands for Examination of Kinds in Natural Diversification and Speciation. Wow, that's a mouthful. Let's take it apart a little bit at a time. Okay, we're biologists, and we want to examine things. That makes sense. But some of you may be asking, well, what do you mean by kinds? I'm glad you asked. In the book of Genesis, we are told that God created plants with seeds according to their various kinds. Then later, God created creatures that live in the water and creatures that fly on day 5. And again, it says that they were created according to their kinds, or according to its kind. There's minor variation in the wording. Then they were blessed, and they were told to increase in the sphere they were created to inhabit. In other words, creatures that live in the water were told to increase and fill the waters. And creatures that fly were blessed and told to increase and fill the earth. Then on day 6, livestock, beasts of the earth, and creatures that move along the ground were created, as you may have guessed, according to their kinds. So when we're thinking about what we're talking about when we say kinds, we're thinking about how God created different kinds of plants and different kinds of animals. So they have a common creator, but they were created as distinct groups. Okay, so let's look at the idea of diversification. Let's look at a plant, Brassica oleracea. Here's a picture of that plant. It's called wild cabbage, and this is cabbage, same species, and you have collard greens, and you have kale, and you have broccoli, and you have cauliflower and Brussels sprouts, etc. Every single one of these are the same species. Do you see all that diversity? That's amazing. And that diversity happens to be very beneficial to us as human beings. It provides us with a wide variety of foods we can eat. It is very beneficial agriculturally. It's interesting too. Okay, let's look at an animal. How about dogs? There are hundreds of dog breeds, and those hundreds of dog breeds have arisen in hundreds of years. Now let's look at that diversity. If you look at the hair coat, what do you see? You see variation in length, in color, in texture. What happens if you look at the ears? There's variation in the size and shape of the ears and how they're held. You can look at the tail and limb proportions and muzzle proportions. There's a lot of diversity in dog breeds that has arisen in only a few hundred years. Now, some of this diversity is practical to us when it comes to working dogs. They're very beneficial. But a lot of this diversity is just something we as humans enjoy. It brings us pleasure to see all this diversity in dogs and to have them as our friends. And it's it brings us pleasure. There's diversity in cattle breeds. I happen to like cattle. And there are thousands of cattle breeds that have arisen within thousands of years. And you have similar types of patterns as far as differences in coat color and differences in other traits. We found that we can breed cattle to produce enormous quantities of milk. Or we can breed cattle to produce enormous quantities of meat. A long time ago, they would have cattle that did pretty well at both, but not as excessive, not as specialized as what we have today. And they'd also plow your field. So there's a lot of variety in cattle. A lot of it is useful to us agriculturally, but a lot of these cattle are also locally adapted. So there's some cattle breeds that are very adapted to dry and arid conditions, and other cattle breeds are adapted to other conditions. So some of this diversity involves adaptation. Lest you start to think this is only in domestic species, let's take a look at other members of the family Canidae. Canidae is the family that has dogs and wolves and coyotes and foxes. And we believe that they're all related. The dog probably descended from the wolf. I've known dog-wolf hybrids in my life, and there's other hybridization that can occur within the group. But take a look at this diversity. You see foxes that are adapted to live in the desert, foxes that are adapted to live in the Arctic, and foxes that are adapted to other situations, and the coyotes and the wolves as well. So you see a lot of adaptation. And this doesn't seem to be unique to dogs, because if we look at hares, you can find hares adapted to live in the desert and hares adapted to live in the Arctic. And there are some similarities in how they adapt. So one of the burning questions I've asked is, how do they do this? How do you get all this diversity that keeps showing up? In school, I was taught the secular view, which probably all of you were as well. And it says that random mutation plus natural selection has given us all this diversity we see today. As a creationist, we have recognized that the Bible is true, and so God created things, so there's created diversity. And there's two ways that there's created diversity. One, because God created different kinds of plants, and God created different kinds of animals. But even within a created kind, you can have diversity. When God created Adam and Eve, he only created 2 humans, but in their chromosomes, each one was paired. And so there could be diversity between the 2 chromosomes. The gene on one chromosome might be slightly different than its matching gene on the other chromosome. So you had a difference of male and female, but you could have other created diversity that was there as well. But after that, we have tended to assume that random mutation and natural selection are how we got all this diversity that we see today. So that I have question marks by that, because even though initially that is the way I explained things, the more I looked, the more I got suspicious that this is not what's going on. When you have 2 views, it's important to go back and look at assumptions. So we're going to look at these at worldviews, and we're going to look at them from a metaphysics standpoint. The secular view assumes there is no God, or at least not a biblical one, who created miraculously. I've been told by evolutionists, oh, you can still believe in God. It's like, why would I want to believe in a God who doesn't do anything? In the biblical view, we have an all-powerful, all-wise, benevolent God as described in the Bible, and he's the creator of everything. These 2 worldviews also have a different history. The secular view has an imagined history based on the rejection of Genesis. Their history was not observed. They believe that life arose by natural processes, and they believe in universal common ancestry. Lest you think I'm being too hard on the evolutionists, I'll give you a quote from a very famous evolutionist, Ernst Mayr. He says evolutionary biology, in contrast with physics and chemistry, is a historical science. The evolutionist attempts to explain events and processes that have already taken place. Laws and experiments are inappropriate techniques for the explication of such events and processes. Instead, one constructs a historical narrative consisting of a tentative reconstruction of the particular scenario that led to the events one is trying to explain. So that is a mouthful, but what is he saying? Do you notice he says instead of using experiments like we're used to in science, one constructs a historical narrative? Well, what does that mean? It means you make up a story. You make up a story. It's consisting of a tentative reconstruction of a particular scenario. In other words, it's a story that tries to account for how things happened, but no one ever observed it. So, if you are told dogmatically that humans descended from apes, and you ask, well, who observed that? The answer is, no one observed that. It is a story that was made up. Okay, the biblical view we've discussed, it has a history in Genesis, and life was created according to their kinds. And that means there's limited common ancestry. Excuse me. So, for example, with dogs, it doesn't bother me at all, since dogs and wolves and a number of other canids can hybridize and have offspring. It doesn't bother me to suggest that all of the family Canidae are from a one created kind, and they share common ancestry. I don't have a problem with that suggestion. You have a very similar body plan there. But if you're going to suggest to me that the hare that lives in the desert shares common ancestry with the fox and the dog, I have a problem with that, because you have a fundamentally different body plan in the hare compared to a dog. There are profound differences anatomically and physiologically. I don't buy that they share common ancestry, and the biblical view in Genesis fits with that. So there's limited common ancestry, not universal common ancestry. A lot of people don't like that I use the Bible when I'm trying to understand the world around me, because they claim it is biased. There is an implication that science is unbiased. So let's address that question. Is science unbiased? Well, scientists are human, and all humans naturally have biases. Ideally, we want to be as unbiased as possible so we can clearly understand the world around us. If we were omniscient, we wouldn't have this problem of bias, but only God is omniscient, so only God lacks bias. But we want to get rid of our biases as much as possible, so we want to understand our assumptions. And using the Bible's history, you can claim introduces a bias. Okay, but if that history is true, it biases us toward the truth rather than away from it. Excuse me. Okay, so what am I saying? I'm saying if the Bible's history is true, then if I use that history to come to conclusions, the conclusions I come to are more likely to be true and sound. So it's going to bias me towards the truth. It doesn't guarantee that I will always have the right answer, but it biases me in the right direction. Rejecting the Bible's history introduces a bias and doesn't even allow one to consider the possibility that the Bible's history is true. So, no, science is a human endeavor and will always suffer from bias. It's not unbiased. It's not like God's word, which is unchangeable truth. Okay, as I was continuing to ponder the question, where does this diversity come from, I decided to investigate the underlying genetic basis of coat color, because it had been pretty well researched and there was a lot of material out there I could read. One of the genes I looked at was the MC1R, and this is a gene in the DNA that codes for a protein receptor on a pigment cell. And you will notice the different coat colors in those two bovids. And the MC1R was the reason why. Now, I want you to picture in your mind a pigment cell, because those pigment cells in those hair follicles, picture a pigment cell. Aren't you imaginative? Yeah, it's called a melanocyte, but it's a pigment cell. And the receptor that is coded by this gene is kind of like a switch. Okay, it's kind of like a switch. It's on the cell surface. It doesn't really look like that. This is a two-dimensional representation of it. Each of the circles represents an amino acid in the protein. And you find the blue is on the outside of the cell, and it travels through the cell membrane, which is in gray, and then it loops into the inside, which is your orangish color there. And it travels back and forth through the cell membrane and is embedded in it. Now, this is only two-dimensional, but it's a lot closer than an actual switch. But for the sake of discussion, we can still think of it as working something like a switch. When there is no signal, the cell produces a lighter pigment, and it can be kind of a golden brown or even a little reddish. It's going to be a lighter pigment. Well, there's a signaling molecule that can come in, and it will essentially flip the switch on so that the cell will produce a darker pigment. So in these two cattle, what you get you would think has to do with the signaling, but that's not exactly what's going on. Mutation, which is a change in the DNA sequence of this gene, affects the receptor's response to signals. So the bovine on the left has a mutation so that it doesn't matter how much you signal, it only produces a lighter pigment. And the bovine on the right has a different mutation so that no matter what's going on with the signal, the receptor is kind of stuck like a switch in the on position, and you always get the dark pigment. Because in most animals, the wild type, you can produce both pigments, and the signal normally comes in at a certain point, and it produces a dark band on a lighter brown. That's typical of the way it would work, but these two cattle have mutations. Well, you might ask me, how do you know it's a mutation? And that's a very fair question, because the biblical worldview has created diversity. Even within a kind, there can be diversity. But there's also a genetic bottleneck at the flood, which we didn't mention, but it is in the book of Genesis, and that affects our thinking, because clean animals and birds were preserved by sevens, but unclean animals on the ark were only a single pair. And this places clear limits on the diversity found in animals today. For example, let's imagine the ancestor of our dogs. Maybe it looks something like a wolf, who knows? But it was some sort of canid on the ark. Those are unclean animals, and there were only a single pair, just a male and a female brought on the ark. Now, they each have pairs of chromosomes, so the gene on one of those chromosomes may have been slightly different from the gene on the other corresponding chromosome. So you can have a few different versions of the gene, which are called alleles, that come through, but only so many. So the best way to know that something is a mutation is to compare offspring to the parent. If we sequence the DNA and we see that the offspring has something that isn't in the parent or either of the parents, then we know that it's a mutation, assuming we've done everything correctly. A new trait is sometimes from a mutation. A white horse would be an example. There is a different gene involved in coat color. Actually, there are hundreds of genes involved in coat color because it's a very complex pathway. And in white horses, it's typically a mutation in the KIT gene. And so if you have a white horse that did not have any white horses in its ancestry, it'd be a pretty safe bet that you have a horse with a mutation in the KIT gene. But you need to be a little cautious with that and we'll talk more about that later. Disruption of a pathway. Well, that's what was going on with the MC1R. You had something going on so that the protein was not responding to its signaling molecule the way it normally would. Isolated. Mutations tend to be rather isolated. So if you just find something in a small group of animals that seem to be related, it may be a mutation. And too many alleles. Okay, alleles are versions of the gene, and they aren't always identical. So If you have 2 chromosomes, the gene if you have 2 of each of your chromosomes, if your chromosomes come in pairs, so the allele on one of those could be different from the one on the other. They could be the same too. So when I was trying to answer the question, where does all this diversity come from and why does it seem to just show up, I looked at more than just the MC1R. I looked at 3 different genes involved in coat color, variable genes in the dog. Some of those have to do with hair length and texture. I looked at genes associated with high altitude adaptation in mammals and genes associated with loss of spines in sticklebacks. So I have looked at a lot of them. And you know what? Are there patterns? Yes. So the first pattern I notice is the genes, and sometimes they don't actually code for proteins, so you'd say technically they're a regulatory region, but they're designed so they can change. So you can do about anything you want to the MC1R, and you don't kill the animal because of where it is in the pathway, in that complex biological pathway. Because of where it is, it's designed in a place where it can take change and you still have a viable living animal. The mutations seem to appear under particular conditions. Domestication is one time where you see some of these mutations, and when it's useful for adaptation. Certain types of genes seem to be affected. For example, certain types of receptors, and MHC stands for major histocompatibility complex genes. This is a number of genes which are involved in the immune system. For any one of them, there are often hundreds of alleles. It's almost as if things were designed so that they would remain variable for the sake of the animal. All of these are involved in helping the organism as it interacts with its environment. Environments can change. And so it appears that God designed organisms so that there can be changes in the gene to help them. And the type of mutation, SNP, is essentially when you have one nucleotide changing. So if you think of the nucleotides in terms of letters, one letter change. Indels are insertions of a segment of DNA or deletions. And then you have copy number variants. So there's this segment of DNA that's repeated a certain number of times. So the type of mutation sometimes varied depending on where you were talking about. But let's back up. Let's look at overall patterns. The big picture is that these mutations adjust what exists. So it requires everything to be in place from the beginning. You had to have the very complex biochemical pathways that are involved in pigment formation before you could adjust the pigments. They may make substantial changes. For example, they often remove the protein from its normal control. We saw that in the MC1R. It made it so it didn't even respond to the signaling molecule anymore, or they can otherwise change sensitivity to a substrate. Now, not so much in humans, but in certain birds and mammals. There have been changes to hemoglobin to help them adjust to high altitudes. Changing the hemoglobin will change how tightly it binds to oxygen. And the ideal grip, so to speak, of hemoglobin on oxygen varies. At sea level or close to it, it's what we normally have in our bodies. But if you go higher up, that grip can make it so you have a harder time getting oxygen from the lungs to the cells. And so some mammals and birds have changes to the hemoglobin which are adaptive. There is something missing in this pattern. The type of changes that build the complex biological networks from a simpler form. You're starting from complexity and you're adjusting it. You are not building complexity. Well, actually, a lot of this research was done previously, and so I felt like we have a lot that's here that we need to understand better. So in 2016, Dr. Kevin Anderson, who was then the lab director for the Creation Research Society, and myself, I was a board member at the time, initiated the eKinds project. And the intent was to get other researchers involved. So, excuse me. We had several questions. How did we get the wide variety of today's species from the small number of animals on the ark? How do new species form? And how does this fit within biblical creation? And can we trace the spread of created kinds from the ark to where they live today? So we have just barely started towards answering those questions, but we have 11 full-length journal articles. The ICC I have not seen published yet, but everything is in for that. So by the end of this year, 11 full-length journal articles have been produced, and we had several other researchers. So it's been highly successful. Well, one of the things I did was I started posting our articles on ResearchGate, and at that time, you could put a hypothesis in. Now, I had two I put in. This is the first one, and it really is more of a prediction. Notice the prediction I'm making. Many adaptive mutations are not the result of random genetic errors. Instead, much like mutations involved in antibody formation, there are enzymes and mechanisms, for example, nucleotide sequence motifs, that guide the process. Well, I had that up for a little while, and then last year it was confirmed. So Monroe et al. Used this little plant you see over there on the right and said mutation bias reflects natural selection in Arabidopsis thaliana. So they were able to control the experiment so that natural selection was not operating, and they looked at mutations and where they occurred. They found that mutations are more common where they're potentially helpful and much less common where they can do harm. So the pattern we see is actually from a bias in the mutations, not random mutation. And so I conclude if it looks like natural selection, but it's not, it's evidence of God's awesome design. So when we look at the comparison of the secular view versus the biblical view, random mutation and natural selection was heavily promoted by evolutionists because it seemed to get rid of a creator. And you know what? You can't explain biology by getting rid of a creator. So for the biblical view, I think we need to replace random mutation and recognize there's design in mutations. That does not mean that you don't ever have errors in copying, because you can. It does not mean that every single mutation is necessarily from design, but it means that, generally speaking, that is what's going on. Okay. Now, what about natural selection? Well, natural selection is supposed to affect the frequency of traits in a population. If you get a mutation, you have one animal with it. What if that mutation is helpful to animals in a particular environment where that mutation has shown up? What if it's helpful? How do you make it so that more animals in the population have it? Okay, I'm going to back up a little bit. Not everything is genetic, so I need to preface what I'm going to say with this. There's a lot of physiologic and behavioral adaptation that goes on. If you start an exercise program, your body will change because it's responding to what you're doing. And so that's physiologic adaptation. If you fly to Quito, Ecuador and get off the plane, at first when you get off, you're going to be breathing heavier and your heart rate's going to be faster. You may notice for the first week or two, it's really hard to climb stairs there compared to what it was at home. But your body will continue to adapt physiologically until the point of 2 weeks later, you're going to have more red blood cells circulating, and that's going to help a lot. At one time, blind cave fish were, that's evidence of natural selection. But it's found out that now you can take surface cave fish and you can put them in an environment, and in the individual that gets moved, you have a physiologic change. So the traits are changing even though the genetics underneath aren't changing, at least not initially. Maybe in subsequent generations they may, but initially they're not. What can affect the frequency of traits in a population? Well, you can shuffle the created diversity around, and we'll mention more on that in a minute. Founder events are where you have like a pregnant female or a small number of individuals invade a new place, and they set up a population there, and they do not interbreed with others of their kind. And whatever traits were brought in by the founders are the traits that are going to be common in that population. Now, natural selection can operate, but before we give it too much credit, genetic mechanisms that bias what is passed on to offspring also exist. They need to be considered. And this is called transmission distortion or meiotic drive. Transmission distortion is a general term. It often occurs during meiosis, so it's often termed meiotic drive. All right, we got to go back and review a little biology. Now, chromosomes come in pairs. We already discussed that, and you would have more than one pair, but here for simplicity's sake, we're going to just show one pair of chromosomes. There is a chromosome that was inherited from the father, the paternal chromosome, and one from the mother, the maternal chromosome. When you're going to have the next generation, the DNA will replicate, and then you get the chromosomes like you're used to seeing them, the little X there. But in order to divide them, you're going to separate the chromosomes so that you only have half to send to the offspring. And they need to line up so you can properly separate each pair. In this case, you see in the third circle down, there's crossing over taking place. So basically, these chromosomes that are the corresponding chromosomes will get connected to each other, and things like crossing over occur. So when you're done, you have separated those two chromosomes, but the paternal chromosome isn't quite the same as the paternal chromosome that was inherited because crossing over has occurred and changed some of the sequence. And the same for the maternal chromosome. This is basic meiosis. Hopefully it's familiar to most of you. Well, this is called homologous recombination that occurs during meiosis. Crossover is the one we just showed, and you swap segments. So you don't have anything particularly new, but you swap what's on one chromosome with what's on the other, the corresponding chromosome. There's something else that can happen, and it's called gene conversion. With gene conversion, you copy a segment from one over that of the other. So you're not swapping, but you're making a duplicate. You're copying over. It recruits some different enzymes. Now, both of these start by enzymes that cut the DNA, and you have a number of enzymes that are involved in the process of handling the DNA, but because you have a different outcome, there are some different enzymes that are recruited for this process to happen. And it appears to be more common than crossovers. It appears to usually involve shorter segments of DNA, but to be much more common. Well, okay, that wouldn't mean much if the chance of copying one direction is equal to the chance of copying the other direction, then it's not going to change our expectations. But there's something called biased gene conversion. When one allele is preferentially copied over onto the other. Okay. So let's take an organism, and they're a heterozygote. So they have two copies of a gene. One came from their father, one came from their mother, and they happen to be different. Well, we would expect normally for there to be a 50-50 chance that they'd pass either one of them on to the offspring. But what happens if you find that twice as often the big A is passed on compared to the little a? Well, when you see that once, it's not a big deal because that could happen by chance. But when over and over and over you consistently see that the one is preferentially passed on to the offspring compared to the other, that's a transmission distortion. One of the mechanisms that can result in this is biased gene conversion. So I had a second hypothesis, which was really a prediction that I put up on ResearchGate. And I said that mechanisms that bias allele transmission, such as biased gene conversion and other forms of meiotic drive, will eventually be shown to contribute significantly to the propagation and fixation of adaptive alleles. I feel like design has been explained away by the evolutionists. And this happens in a lot of areas in biology because, some things would imply a creator. They have assumed that Mendelian inheritance is always normal. And that means in the heterozygote, where there's two different alleles, they are supposed to pass it on with a 50-50 chance that one of them is passed on to the offspring. And it's been explained why by calling all else intragenomic conflict or selfish, so as not to imply that there might be a purpose. So if you read the biological literature, you will hear about selfish genetic elements and parasitic DNA and so forth. And that's a judgment they have put on it. And I feel like that really, it doesn't just explain away design, it also isn't really helping us look for what's going on. Design recognized. Why do I think I see design? Because meiotic drive is relatively common, and there are several different forms of it. Serious problems associated with it are rare, though there are examples where there are trade-offs. And these processes are regulated by various enzymes. Speaks to me of purpose, speaks to me of design, speaks to me of a designer. So when we're comparing the two views, it's not that natural selection can't occur, it does, but it's typically been given amazing powers that are far beyond anything we could do with artificial selection. And since that's my background, I started recognizing, wait a minute, this is not realistic. Natural selection can occur. It can be helpful or not. One of the conclusions of the grants as they worked on the Galapagos with the finches is that natural selection does not work the way Darwin suggested. And I suggest that in general, while natural selection can occur, I think we're going to find, and this has not been confirmed yet, it'll take a while to be confirmed, that there is design in what gets passed on to the offspring. If it seems best, if it's a situation where a 50-50, chance of getting an allele is there in a heterozygote, then that's the mechanism that'll go on. And if it is advantageous for it to be different than that, then that's what's going on. So I see awesome design there. Okay, ekines. Using the Bible's history in Genesis allows for a more accurate understanding of biology, allows for more accurate inferences about natural history, and highlights the manifold works of God. O Lord, how manifold are your works. In wisdom you have made them all. The earth is full of your creatures. Thank you.
Thanks for joining us for this video. Please feel free to share your thoughts in the comments below, including any questions you would love to see us address in future videos. If you're interested in more information about Clarks Summit University or our biology program, click the link below and we'd be happy to help answer some of your questions. CSU biology majors prepare for careers in medicine, research, biotechnology, and further scientific education. With plenty of hands-on laboratory experience and opportunities to lead in the lab as an undergraduate teacher's assistant, students understand the beauty and order of creation through a biblical worldview. And as we stated already, our mission here is to develop Christ-centered and career-ready graduates. We hope that you have a great day, that you've enjoyed this video, and that we'll hear from you soon. God bless.